All right. Well, let me. I guess we'll start it. We're starting DARPA time, so which is according to our clock right now. My name is Mark Lynn. I should probably start off and say that I'm actually I'm not a government employee. I'm a contracted. I do contracting support for DARPA, so I can't answer a lot of the questions that are on behalf of DARPA. I can answer some general questions. We do have the way DARPA does things is we do have an email address. We'd like to try to distribute all of our information uniformly. So even if you had a question and Colonel Wattendorf was here, you'd probably also direct it to this email address because we want to make sure that if you ask a question, that everybody also knows it. We try to do it as a fact on a website. So here are some cards just listing the email address. I'd also introduce Mike Muddy. He's our contract. He is a government employee, and he's a federal employee with DARPA. So he can answer anything that's more related to the contracting mechanisms and how DARPA works. The reason we're here, and a little bit about my background. Prior to coming here, and some of you know, I was with the NCI at a program called IMAT, and it was a program that was modeled after DARPA. It was molecular analysis technologies. It's one of the few programs at the NIH that focused on milestone-driven, non-hypothesis-based research. And there's a couple of people who actually received IMAT grants that are in this room. I was brought over to DARPA just recently. This interest in molecular diagnostics is pretty new, which I'll exchange and show you kind of what we think about molecular diagnostics and then what our attempts of going in this space is. Because it's new, one of the reasons that we're out here in this kind of small, intimate classroom is because we actually want to reach out to a lot of the academic as well as small businesses. As you know, DARPA is really big into teaming. So we think a lot of the new approaches that people at very early stage research, as well as the ones that are being carried out by industry right now, we want all of them to kind of address a problem that we have that we think is a problem and that DARPA is very interested in trying to stimulate. So really quick background about what DARPA is about. DARPA was formed in 1958, basically because of Sputnik. The USA was caught a little bit by surprise when Russia came with Sputnik, and basic DARPA's missions is try to stay as technologically advanced as possible. And that's kind of what we're trying to do, which is why we're capabilities and mission focused. Our idea is we have an idea of what we want in terms of technology, and we just need to get there. It's very different than the NIH and SF models where it's really supporting kind of the basic science. For us, we can support the basic science, but it's really aimed at getting towards an application. So if there's no one investing in a certain space that is kind of basic science space, the NSF isn't doing it, NIH isn't doing it, then there's a chance that DARPA will do it. If you guys, hopefully the notice, we're also here kind of on more of a one-on-one basis. It's kind of shows a little bit how DARPA is trying to be a little more creative. We're calling it on our side speed dating. I don't know what everybody's calling, but basically it's a chance for you guys to learn what DARPA is interested in. So Colonel Wattendorf, who's flying in right now, will actually give the exact same presentation at this other meeting. But he's also going to meet one-on-one with interested research groups to talk a little bit about what we're interested in molecular diagnostics. Mike Muddy will be able to talk about funding mechanisms. It's much different if you're used to the NIH, NSF. It's much different than what you may be used to. So any questions that you have about how you contract and how you work with DARPA on the financial end, that's what Mike was willing to come out here also for. And then I'll be there as well as the third speed date to if you guys are interested in talking about your technology itself. We have a finite, the projects that we have, there's an end date to it. The reason that there's an end date is because the program managers that are brought into DARPA all have an end date. So they come into, they're not what you're probably used to at NIH, working with a program manager who've been there for 20 some odd years. At DARPA, they have a tenure, which lasts between 2 to maybe 5 years. And they're brought in because they have a dream, they kind of have a mission in mind, an application, and they're able to tell DARPA leadership, and DARPA's basically invested in that idea. So because of that, that's why it's a whole mission focus. So they brought in someone to come up with a specific technology, and we're basically there to invest basic science or the application science to get to that idea. So we do have a finite duration. If you have worked with DARPA, we're very milestone driven. If you do contract with DARPA, you'll probably be used to monthly calls from our technical team to see your status. So there is a lot more accountability, monthly reports, and that's stuff that maybe Mike can talk about, or you could look at other BAAs that we have. We usually solicit everything through a broad agency announcement. Talk a little bit about that as well. And that's how if you want to look at how to work together, because it does comes with the strings attached. So you will get monthly calls, you get monthly reports, and so basically because we're trying to get to that capability that we're trying to get to. Everything coming out of DARPA is very multidisciplinary. So what you'll see is a lot of math team, scientist team, different sciences, engineering, physicists, and it's very rare when it's a single grantee. So when you come into DARPA, and especially the stuff that we're looking at, you have to come in as a very strong team if you're going to come into the solicitation. It's because we're looking at a very specific target and a very specific capability, and we want to see how you're able to integrate all the different teams to get to that capability. And I'll explain a little bit. So this is kind of giving you a taste of what I'll talk about in a little bit. So again, it's very multidisciplinary. We call them performers, grantees. You guys are our performers if we do fund you. And again, we're looking, and as most of you guys know, DARPA is very high risk because we are trying to get to that idea. I mean, the ideas that we have out there are, it could be anything, from the Star Trek tricorder all the way down to something that we could apply really quick. So characteristics of the DARPA program, we are looking for something that's very revolutionary. And coming from the NIH, we're really looking for something that's very different. If it looks like something that can be funded by the NIH, we probably will say, well, you're not really advancing the science in a way that we want it to advance. So we're looking for, what is kind of your best idea that you have, maybe that you don't have funding for, and how would that change kind of U.S. capability? It doesn't have to be all direct military applications. For us, we're a little bit in the clinical space. The Department of Defense has a healthcare provider that's on the order of one of the biggest HMOs. So we are looking at also civilian capabilities. We're thinking about FDA clearance ideas as well. So everything that we're looking at, it really has to be a revolutionary change kind of across the board. Now that's not for all DARPA programs, but the ones that I'll describe a little bit later. We are looking at the civilian market, civilian interest. So if you send us something that's just specific for someone who gets hit in a Humvee that encounters traumatic brain injury, it's probably something that you shouldn't limit yourself to. We're looking for platform capability. Again, everything is the idea of the program manager. DARPA is a very flat structure, and the program manager is the one who kind of sees where your ideas fit and how it fits into getting to what he wants. That specific tool, that specific diagnostic. So again, everything's divided by projects. Very rarely is it investigator-centric. So if you do solicit funds from DARPA, come in as a strong team. If anything, that reach out to everybody that you can think of in industry, within multi-disciplines across the board, and come in as a very strong team. That's probably the biggest recommendation. If it's a small project, it really depends on how we can make it fit with our overall. One thing it's a little bit different because it is run by the program manager. We don't have study sections, so it's very different than the NIH, where basically you can solicit an idea. We do have what we call an open BAA, where if you have any idea, you can do any solicit anytime, and we try to get it out. If it's something that aligns with the program manager, then it's something that we can fund relatively quickly compared to the NIH. NIH may take, as you guys know, a year. For us, we do an internal government-only review of it. Again, it's to give us a little bit of flexibility in getting out funds and getting within that time span that we want. So again, flexible, rapid review and contracting. That's kind of the biggest kind of advantage to working with DARPA is you'll know right away whether it's going to be funded, whether it's not. And again, because everything lies with the program manager, you may think, well, this is the greatest science. How could you think the science doesn't work, this and that. But if it doesn't align with what we're trying to get with the program manager, he's the one who has actual control over it. So the science may be great, but we're not looking at funding necessarily basic science. We're looking at funding a specific capability. So it has to align with that. And that could be something to get back. Unfortunately, it doesn't align with what our mission is. So you may get those types of responses, and that's just the way DARPA works. And we're driven by quantitative milestones. We're very milestone driven. So in terms of diagnostics, this is really our overview of it. Again, what we're here to do is to explain, kind of to get you thinking about what DARPA is thinking about in diagnostics. And we're also very interested in hearing what are your challenges. We want to know, well, okay, maybe you're going in this approach for a certain technology, for a certain space. If you could go make it towards a diagnostic capability, and what's really your potential? How widespread could this be used? So everything that we've described here, we're looking for really platform approaches. We don't want to invest in a specific technology that only works for one disease condition. We're looking at something that you could really get something that's hopefully FDA cleared, and my colleague from the FDA is here, and he'll talk a little bit about it. But everything that DARPA uses, the military uses, has to be FDA cleared. In order to get it FDA cleared, DARPA doesn't want to be your only investor in getting this technology out. With a lot of many of the DoD technologies, DOD ends up being its only customer. We're actually hoping for us, in terms of our big vision, that you actually have a commercial market where you can get funding for. DARPA can basically de-risk everything in terms of getting that up to that stage, which is why we're looking for that team integrated approach. You're able to develop, hopefully, and maybe Daya will tell me that this is too dreamy, but we're hoping that you're able to develop the technology within a commercial clinical context, get FDA approval for use in a certain, and if the military needs to apply the technology to something that's more military-centric, that we can basically buy down that technology, and it can get FDA approved, because FDA has a predicate type of, at least, device that they're comfortable with, and that basically all they have to do is evaluate the science in terms of the biomarkers that we're using in this new device. So we are looking at platform capabilities. We're looking for that end-to-end approach. We're looking for something where a biospecimen comes in for all of these. Biospecimen comes in, nobody handles it. Comes in for biospecimen, and the data comes out. And it's really within context of when a physician and patient interact. So you may look at it and go, well, is this really a DARPA challenge? We actually, the more and more we talk about, the more and more we do these market surveys and kind of look around, it ends up being a big challenge that we think needs to be addressed, which is why DARPA brought in Colonel Wattendorf. We just started in May. So we don't have any active solicitations in this, but let me walk you through what we're thinking about. And again, these are just ideas that are thrown out there. We'd love to hear your ideas. Right now, it's a great opportunity to talk with us, to refine our ideas, because we don't have any solicitations out right now. But that may change. But that's why we're here to do a kind of this market survey. We're hearing your technical challenges, and then see what can come out of it. So the first one's looking at the point of need, which is the one on the leftmost, and that's really what everybody thinks the pregnancy, being able to do something just like I could. When you're at home, you can test by yourself. There's nothing that requires phlebotomy, and the user can do the administration themselves. So for the military, you could use it out in the tent, but also at home you could use it as well. And basically, the readout is go see clinical attention. So if it's a soldier that's out in the field, they go, okay, we need to see a medic, or we need to go to a hospital, or what we call combat support hospitals. For anybody here in the civilian side, state side, basically they can go see a physician. And that's why we're looking for stuff that's CLIA waived as well. And if you guys aren't familiar with the CLIA waiver, you can see it's popping up on here. Those are things that we're going to be looking for when you do propose, because we are looking for CLIA waived capabilities, which allows somebody to do the test at home or within an office setting without being in a reference lab. The second one we're looking for is something that you could use within an office setting. So this one's really kind of a single disease type effort. The second one is a little more multiplexed. Again, it's more important that the answer is generated within what we call the physician-patient encounter. So a physician knows right away there's nothing that gets phlebotomized and has to get sent off to a reference lab. But hopefully the physician can actually tell what types of diseases right there, what types of conditions are. So minimal medical training, so anybody could really administer these tests. It still could be at home or an office or a clinic, or even a medic could do it. A little bit more multiplex. This increases in multiplex. If you see simplex, multiplex, and highly multiplex, you can be able to follow the data and also be able to act on it right within hopefully that 10-15 minutes that they're seeing a patient. So the actionable turnaround time is the same thing, right there. And again, we're looking hopefully at something that would be approved by the CMS and by the FDA for CLIA waiver. The third one we have is the central lab kind of capabilities. That's something that I think a lot of people can address. We're looking for still minimal operator interference. Basically, someone could just insert a biospecimen in there. This can be with phlebotomized specimens. We're hoping that these don't require phlebotomy or extensive sample preparation outside of the box. That one we're looking at the next. If you talk different crowds, multiplex can be many different things. It could be you have a lot of controls, a lot of negatives, false positive, false negative controls within the same device. We're looking for that. We're looking at multiple diseases. We're looking at ways you could look at co-infection. So if somebody has definitive interpretation against, it's kind of obvious. All these we want to have a definitive interpretation, and we have an idea which we'll talk about in the next. And the next slide, which talks a little bit about how we think you convert something from a clinical diagnosis specimen to something that is more that we could possibly use for biomarker research to also be able to kind of close that loop. So kind of the common attributes across all of them, FDA approved. So everything should be built within context of getting an FDA approval. Hopefully, maybe Dia will be the one to judge on that. Again, integrated everything, biospecimen in, data out. Platforms, self-collected biospecimens are the best for us, except for the last one, obviously that's where it's getting shipped. Multiple biomarker classes. We're not looking for a new glucose meter. We're looking at stuff that's really looking at the new nucleic acids, all the omics. Wireless transmission of data is something that we're beginning to look at. Is there a way that you could, if you do this point of need device at home, that you could send the data from that device to a clinic so a physician can look at it in real time and say, yeah, actually, you don't need to come in right now, but we want you to test every hour. And let me look at the results after a 4-hour duration, and let me tell you whether to come in the clinic or not. Or don't worry about it, take an aspirin, take whatever. Or they can track the course of disease, make sure the therapeutics, it's kind of this whole personalized. But the wireless transmission of data is something that we are trying to work on from both the policy side as well as the scientific side. And then centralized interpretation, distributed processing is required. That's also another idea that we have, especially for the home point of need idea. That basically, you have something the equivalent of a BlackBerry. You test yourself there, but the onboard doesn't actually have any analytic capabilities on. But somehow all the data, all the analytics get shipped wirelessly to a centralized lab. And this is something just to get around, hopefully, the CLIA waiver capabilities, where you have a dedicated and a trained clinician that can look at the raw data and give a diagnosis from a remote site. So that's something that we're looking at in terms of can you centralize, and we're working on the policy side. You may think there's no way the FDA is going to approve that, or that, FCC, but it's something that we're actually trying to address as well. Again, we're, for us at the DARPA side, we're trying to make sure everything has a clear pathway. For you guys, we're looking at can you build the right scientific teams to go towards those ideas, to what we want in terms of enhanced capability. I hinted at this kind of integrated diagnosis enabling research, and this is an idea that we're kind of shooting around and seeing what's out there, but right now we're looking at something, and it's all kind of just in the imagination space right now. What we're looking at, it seems like a really simple idea. It's a dried biospecimen card. You guys may be familiar with the filter cards that are used, where it's dried blood spot cards. If you guys have seen any news, a couple weeks ago they had an issue on it. And at the same time, they also talked about a lot of problems with using these paper-based cards, especially for next generation sequencing. And we've talked to a couple from the people that have written these articles or that were interviewed, and they said, yeah, we can't even use them in mass spec because of the chemistries and everything. And the cards have been around for 50, 60 years. So we're looking at something a little bit different. We're looking for what we're calling kind of this dried biospecimen collection matrix. The idea, if you just kind of, without looking at the slides, if, say, we'll kind of go in the military context. If you have a soldier that's outside and he thinks he gets exposed to something, so there's a time, he could basically at that time be able to spot his blood and capture whatever, theoretically, whatever biomarkers were present in his blood that maybe in response, whether it's a metabolic change system, or maybe it's even a sign, an actual pathogen. He actually spots it and preserves it. Now we have a card that is both geospatially mapped, because you know where he was and what time it was. So if we have 20 people who maybe encountered an exposure, now we have 20, we have kind of a bigger cohort to study with. Now all these soldiers will carry in this card, and we'll go over and they'll give it to the medic possibly, and put it maybe in a point-of-need device. From that, they're able to analyze that and say, no, you're clear, or you're not clear according to my device. But unlike most point-of-need devices, the specimen isn't tossed away. Right? We actually still have a card that has a specimen. So what we can do is we could send this card over to the central multiplex analysis to go over there to the central labs and really do more thorough analysis and see, number one, maybe we find out later that these soldiers are becoming symptomatic of something that we still don't know. It's the same cohort of 20, but now they're starting displaying some type of symptoms. Can we use it now that we have a card where it has, you know, for all the DoD, we have all their electronic health records all in one Oracle database, so we know family history. We now also have a card that has this geospatially mapped, that has time and what could have happened, as well as a cohort. So we kind of have a good way of doing it. It's an interesting experimental design question in terms of a biomarker discovery and validation effort. But more importantly, we have the specimen that can be easily shipped off of a card, so at the end they can just batch them together and send them over maybe to the CDC or somewhere at stateside, or maybe at one of the bigger capabilities. So that's kind of what we're envisioning in terms of the dry. We're not looking for the next paper. We're looking at something that's a little more compatible for the next generation, you know, proteins. You know, is there a way, if you spot right now on the current dried biospecimen cards that are out there, the proteins are dried down, they get denatured. And they also get, when we talk to the pharmaceutical industry, they also get derivatized by the chemistry that's on there. Is there a way that we can preserve a protein in its native state within some type of a card, so that we can take it off and actually do not just a mass spec identification of a biomarker, but maybe we could do some kind of functional proteomics, some kind of activity-based assays, and we could actually learn a little bit more enriching for a biomarker discovery effort. And we could also maybe validate, because maybe we have 3 different spots that we can work on. So simple biospecimen collection without medical training, something a soldier could carry just in their pocket. You know, if they get exposed to something, we tell everybody to spot yourself just to make sure. It could be a burning oil field, or it could be some kind of intentional, you know, exposure. You know, potential direct analysis for immediate diagnosis. So again, we could integrate it with this point-of-care device. So medic could right away clear them if we do have that capability on that kind of simple device. Simple to transport. Can we just batch them up and FedEx them all over? Even if we're just going to archive it, maybe 20 years later we find out that there was something that everyone was exposed to, and we can still do some biomarker research on. So we're looking at long-term stability. And we're also looking at not just blood, but our other different parts. So, you know, again, this is kind of our dream, and we'd like to know how you think your best ideas for getting to that. You know, DARPA kind of dreams big, and you guys may think, you know, this is kind of crazy, but we'd love to hear how you think we could get here. So we're looking at blood, we're looking at urine, something simple like saliva, tears, sweat, even possibly feces for information. How would we do this? Would we have multiple cards? This is what we'd like to throw you. Do we need multiple cards by nucleic acids, by protein, or do we need one per specimen? How complex can we make it? We prefer different answers. So that's kind of what we'd like to hear from you, either through our open DAA process or through these kind of open conversations, which is why we're reaching out to everybody here. And again, you know, time and location annotation, simple to carry. Lossless preservation of analytes, so the biomarkers are hopefully preserved for that analysis. Without knowing how it's going to be analyzed, maybe it's technology 10 years later that can analyze it. We still would love to have that card. And it's universal to multiple, so it's not dedicated only to my machine, but you know, could be used for different approaches. So to find out more about DARPA, we do have you know, the DARPA.mil website. If you go, so we're out of the Defense Sciences office. There's 6 offices at DARPA. Defense Sciences tends to be a little bit more of the physical science, biology. It's very basic science oriented. If you go on the DARPA website, you can see how all the different offices are laid out. Some of them are very MEMS oriented. for us, we're very basic science oriented. Out of the staff of maybe 20 or so program managers, about 6 of them do have MDs. So, there's a little bit of a clinical push towards it. We do announce our solicitations through kind of 2 different forms. One is on our website, so if you go here, there's a little solicitation box. Keep in mind that for DARPA, because our programs have a short lifespan, it's not like the NIH where you'll see a program come up and there's set receipt dates. We'll announce it, it'll be out for 45 days, and it'll be gone in the ether, and you can't respond to it. So occasionally check it, look out there. You know that there's an interest in diagnostics. That's why we're out here. That's why Mike's willing to come along with us to come out here. So I'd say probably in the next couple of weeks, I would continue to look at this solicitation website for different ideas that we may have. The other place that we announce is Federal Biz Opportunities. That's a little bit more of a tangled web. If you're looking specifically at DARPA, each office has that little solicitation site, and I would look there. We're also trying to explore a bit more and see, because we are a science-driven organist, we'll actually transition to our colleague Daya. We just held a workshop just a couple of weeks, 2 weeks ago, 2-3 weeks ago, with the FDA, just looking at the capability. What is the potential for having something where you're able to continuously monitor someone's physiological status in real time? So something that we'd love to just hear ideas about. The idea is basically, what happens now if you're not looking at a snapshot of somebody's health, but you're actually able to track period and you add time as a parameter to biomarker. Now you're looking not just at infectious disease, but you're looking at processes. Can you look at healing processes to see how fast someone's healing versus someone that's not, and how does that change how medicine's done? Again, this is just to explore what's out there. We work with the FDA just to make sure that, again, you give us the good science, tell us how to do it. We're going to work as hard as we can on our end to make sure that it goes to the FDA. DARPA, in a way, acts as venture capitalists. We're very early stage technologies. We'll try to sell it either to the source, to get it commercialized, or go within the different branches. Because we do want to see, if we do provide an investment to you, that we do want to make sure it actually comes out at the end. So that's kind of my talk. Again, we are here for one-on-one meetings. It filled up a lot quicker than we thought it would. So what we actually did was we locked a lot of the times down, because we didn't want to come here and go, well, we're here, but sorry, you can't meet with us. So at 10 a.m. tomorrow, we're going to release a bunch of times for you guys to go and sign up for the one-on-one meetings. Again, it's kind of the speed dating concept. You'll meet 10 minutes directly with Mike. He'll talk a little bit more about the contracting mechanisms that we have. Dan Wattendorf will be here. We'll talk a little bit of kind of basically what I just walked you through, and I'll be there to kind of look at the technologies that you may have. So if you want 10 a.m., we're doing it tomorrow because we're doing the same talk at this conference. But at 10, they do get opened up. So if you are interested in meeting with us, we're at the Hotel Palomar, which is downtown San Francisco, kind of right by the Old Navy. That's down there by Union Square. And we're going to be at QB3 UCSF on Friday in the morning. And that's kind of all I have.
Yeah, a couple of, I want to add to this chart, obviously. There's a thing called grants.gov, where if we're anticipating grants, we put it into grants.gov. We haven't decided if we're going to issue grants or not yet. And then what I'd recommend is go to this website and look at the solicitation. The DSO, the Defense Science Office, broad agency announcements, are pretty formulaic. So while the technical thrust of a particular BAA will be unique to that BAA, the boilerplate stuff in terms of all the administrative is in there. It might be a good thing for you to look at if you have any questions for me when we do our speed dating.
Thanks, Mike. And if you'll see the programs out there. If you have an idea that doesn't fit within any of our programs, we have an open BAA. So that's an open. Anytime you can just throw us an abstract. You don't have to submit a full proposal. You could submit a full proposal. You could submit an abstract as a feeler. It'll get sent to a program manager. So my recommendation would be to see what the different program managers' thrusts are, to see if you would even get interest within the Defense Sciences Office. But you can always at any time submit a response to an open BAA. And that's, again, just to kind of say, well, we may not have all the ideas out. Why don't you tell us what your idea is, and let us know. So I'd like to thank you for your time and for making it down. I guess it's just, okay, Naya? And again, there's a card in there. If we do, there's an email address that's on this card. It's the same one that we have on the announcement. It's DARPA SN1115 at darpa.mil. So anybody on the webcast, it's the same one that was on there. You can submit your questions there. If we do, when we have a solicitation out, this email address will change. Again, because we try to, if you call and ask a very specific question, we're probably even on the phone not going to be able to give you an answer. We'll tell you email it, and what we'll do is we're going to put it out as a fact. So that's how DARPA works. So make sure, again, any questions between now and any solicitation, feel free to email us. And there's a bunch of cards that are around there. So thanks again for.
That email address will change once the formal DAA comes out.
Thank you, Mark. I'm also going to talk to you about a small roadshow that we also have, and this is a brand new initiative that FDA Center for Devices and Radiological Health has initiated. We actually announced this program early this month on February 8th, and it is one other thing I want to tell you before I move on to next. My presentation is that we have this is an innovation program at the Center for Devices, and we have a public workshop on March 15th. So it is on the DAS website. I have the information about the website too, so which everyone can give their comments to us. So I am Daya Ranam Karachi at the Center Director's Office. I'm a network leader and working with the in-center innovation team, and as the center director, associate center director for innovation is the one who leads this. He's Dr. Jonathan Sackner Bernstein. And so before moving on to anything, I just wanted to talk to you about our mission here. And so you may be thinking that what are the regulators have to do with innovation? So actually, it is to promote public health. So we definitely need to work on innovation. And if you look at what are the challenges for innovation, I'm going to get to that in a minute. But if you look at innovation, what I'm talking about is like why innovation is needed. Actually, there are for medical devices, we have several medical devices that have been tremendously supportive of innovation. Actually, if you look at like coronary stents, drug-eluting, or then also implanted biostensors, these have been really useful in paradigm shifting type of technological advances. So, but still, those technological jumps actually is helping us move on to these paradigm shifting technologies. But what happens is that it takes its own time. It takes its own time to move on, so that because of these challenges. So one is actually these true transformative products bring unique scientific and regulatory challenges that these are actually new devices, new technologies. So when it comes to regulators, we don't know actually what is happening with those things, because these are like new questions, new regulatory questions that comes to us. And then another item is actually a lack of regulatory flexibility. Uh, we are working within a regulatory framework, and then within that framework, actually, we may not have enough flexibility to accommodate this new tool. So these are actually one other challenge that we are facing. Then also lack of infrastructure, lack of infrastructure for rapid development and assessment of novel technologies. This is where actually a device developer may be developing this kind of paradigm shifting devices, but what may happen is that he may not know about how to run a clinical study. So this may be changing, but that is how it works. So we are working in different silos. So if we can address these challenges, that will be really useful for the rapid development of these true transformative devices. So I'm going to talk to you here about only a part of the innovation initiative that we have, But please visit this website for more information. Actually, it is also for comments, and you can also participate in on the March 15th workshop that we have. It's a public workshop, and so I'm going to talk to you about the innovation pathway first. And if you look at this corner here, this is actually the regular development pathway. Right now we have regular development pathway means that these are the places where a device developer can come and interact with us. So we call it pre-ID stage. Pre-ID means pre-investigation or device exemption stage. That's where a device developer can come with a concept and their protocols and all that. We do not look at the data at that time, and then also the next item is actually IDE stage. That is investigation or device exemption. When we determine that okay, this device poses a significant risk to the patient, so it allows you to get FDA approvals and then also IRB approvals, all that. So these are the stages that you get to connect with FDA. And actually, the real interaction happens at the regulatory submission stage, and when this is where actually we will run into problems, because the clinical data and all these data submission happens at this stage. So when the reviewers look at, they may go back and say that okay, we need more clinical data, more design changes. And so what we are trying to do here is that actually this stage we want to bring it up to all the way up there. And so by that, then if you interact with the FDA at that point, then we can all work out almost every single thing. So that's our plan actually. So this is not really a new pathway, but it is a way of a regulatory mechanism that we are developing, because we are all working within regulatory framework. So we have to have public support and all that. So that's what actually we are trying to do here. So it is a special regulatory mechanism for innovative devices. And then so if there are questions, if there are problems that we don't understand, so we are developing mechanisms that we can identify external subject matter experts to. So that's the innovation pathway that we have. And so what are the devices? Then this is also a small change from the previous one. So this shows actually how the device development happens. So it starts from the discovery, ideation, then invention plus and prototyping, and then preclinical, clinical, regulatory decision, and then product launch. So so what happens is that this is where actually the regulators come into regulatory interaction happens. So by this innovation pathway, what we are trying to do is we are trying to move this one to as far as we could. So start interaction from the beginning. And so what are the devices that we have now? This is a pilot program, as I told you in the beginning. And so we have actually accepted one test case for this one. This is what come from DARPA. It's a DARPA funded program. It's a near natural upper extremity prosthetic arm, which I'm going to show you a quick video on this one. So how true transmitter devices that we are talking about here. So here is a near natural prosthetic arm actually to replace restore the arm, fingers, and the hand. So this is actually this each unit, each piece has a like 27-degree motion in any direction. So and eventually this will be controlled by a brain chip that is placed on the brain, which will uh, work with the the neurons based on the neuron signals. So this is the kind of actually devices that we are talking about here, and and then also unmet public health needs, any unmet public health needs which if we can think about, those are the devices that can come. Oops, sorry. Next one. So for this previous one, yes. For this innovation pathway, actually, the we can, how we select devices for this innovation pathway, that and talk to us about this. And then also, when a submission comes in the normal other pathways, then the reviewers can suggest to us, promote suggest, propose that, okay, this is good for a innovation pathway. So that's how we are talking about. But at this time, because of all other programs that we have run it smoothly, so this is kind of like a pilot program right now we are running. So, and then I'm going through the slides quickly, and then there are a couple of more I would like to talk to you about. Also, as part of this innovation initiative, we are talking about advance in regulatory science. So one item, actually one component of that is interagency public-private partnership. So under that one, actually, we have signed a memorandum of understanding, MOU with DARPA, and for actually to have workshops of transformative paradigm shifting device diagnostic concepts. Actually, as Mark said in the beginning, this is one of that we had in the early this month for in vivo bioscience biosensors. So under this one, actually, it was the theme of that was in vivo biosensors for health management. So in the beginning, we asked the participants, how would you see, if, as a technology, biosensors, how would you see this one be useful for health management? So two things came obviously, that is the early detection and therapeutic monitoring. So this is actually just a cartoon showing uh, the pathophysiology of the disease, disease progression. So in vivo biosensors can be helpful from actually palliation, where we look at the symptom and treat it, to prevention. So, but that's our concept. That's where we want to be. But obviously, it comes with so many challenges. So scientific and regulatory challenges. What are the validated biomarkers that we can use for in vivo and under in vivo conditions? So we know that, like Mark said, the glucose sensors, glucose would be plenty, available in abundance, right? But what are the other biomarkers that we could use? Then also biocompatibility. That's a major problem that we have. So when you put it in our body, definitely it is subjected to rejection because of the foreign body response, information and encapsulation, etc. So then also biosensor integration, putting these pieces together. How we could put these pieces together. Uh, so then this another piece that I will talk to you about is under advance in regulatory science, under this initiative, is that promote new tools for device review and assessment. So one is in silico modeling and large scale validation standards effort. FDA already has programs for standards developments in collaboration with ISO and ASTM, which are the institutes that are developing the standards. But those standards, when they are coming to us, it may be actually, we may have review submissions already with us. So we want to advance our program a little bit early. So this is one initiative that we have started to undertake. Uh, that is actually Federal Whole Genome Sequencing Analytical Validation Initiative. This is a massive undertaking that we are thinking about with in collaboration with other FDA, other federal colleagues, NIH and NCI, like that. So the goal here is to develop principles and criteria for analytical validation across whole genome sequencing platforms, and to assemble infrastructure like physical characterization of one whole genome. Okay, it's a medical grade sequence, one whole genome that we want to validate against any genome that comes to us. So if you look at a bioinformatics pipeline, if you are validating if you are taking one genome, which takes about, actually, depending on where the data is generated, where the data, what data we want, what raw data we want, it may be like 10 gigs to more than 100 gigabyte storage. So those are things that we don't know, we have to work on. So these are action activities that we are engaged in under this initiative. So more on this one, actually. So you may know better than me, actually. It is rapidly developing technology under whole genome sequencing, and it has potential to replace all current molecular diagnostics like PCR methods, and then also has a variety of clinical applications in personalized medicine, such as haplotyping, carrier screening, and all that. And then also, but again, we have the same problems coming with scientific and regulatory challenges. What are the medical grade sequence standards that is we can have? We don't have those now. And then validation protocols, and then also knowledge and access to bioinformatics pipeline. Then data management and infrastructure. All these things are big questions to us. But will this, if we don't think about this, it will still, the submissions will still come to us. We have to be ready, and then if we are ready, then when it comes to us, then we are ready too. So that's what actually we are working right now to move forward with this. And then I would like to stop in thing. I just want to mention about this one. Also part of this initiative is that Centers for Excellence, certified clinical test centers. That means, because just for the same reason I was talking about, working in silos, the device developers may not know about, where to go for, to run the clinical studies and all that. So we were thinking about, okay, we can talk about certified clinical test centers, and then with that, what happens is they have to follow certain prerequisites, and there are actually academic centers that can accommodate this, because they have the experience, the device development experience, and also clinical running clinical studies. They accept outside data, foreign, in other countries, but we often run into a problem where the data that comes to us is like not adequate. So the way the studies have been done is not adequate. So the robustness of data is not there, and then also the study that is conducted may not resemble the population demographics that we see over here. So those are the questions that, but so we can try to leverage those loopholes, if you will. And then in conclusion, I would like to just point out these three things: innovation pathways, regulatory mechanism to provide attention it deserves. Collaborative efforts are needed to advance regulatory science on paradigm shifting health management concepts. Already available resources can be streamlined to strengthen regulatory science for medical devices. So this is my contact address, and then I will leave this one for a minute if you have to write down the website information.
Does anybody have any questions for Daya?
Well, I'd like to thank everybody for coming. Hopefully, if you are interested in meeting with us again, the website for the DARPA kind of one-on-one sessions, I think that's probably an announcement that circulated. How you guys found out about this meeting? Right now, if you look at it, it'll be full. So look again at 10 in the morning on Thursday, where we open them up again. Again, you don't have to meet us. We're also very open to phone conversations as well, particularly about learning more about your science. You can email us here at this email address on this card. For those on the webcast, it's the same one that the DARPA-SN-11-15 at darpa.mil. And try to arrange your phone conversation. You could be with, we'd love to hear about what your challenges are, particularly since we're in this pre-solicitation phase before something's actually out in the streets. So, we do want to hear your idea, and again, there's the open DAA as well that you guys can submit anytime. So, thanks again. Hopefully we can see you in the next couple of days or sometime soon. Thanks.
Thank you.